Good afternoon, good morning, good evening, depending on where you are in the world. My name is Jason Acker. I'm a professor in the Department of Lab Medicine and Pathology at the University of Alberta, and I'll be chairing this next portion of part one of this session. So it's my pleasure to introduce Dr. Birgit Glasmacher, who's going to come and really set the stage for further discussions around the practical side of cryopreservation by helping us understand, again, the basics, basic damage, cell mechanisms, cell protection, and really focusing on the stress and cellular events that occur during cryopreservation. So with that, Dr. Glasmacher, it's the stage is yours.
Thank you very much for this nice opportunity to talk for me this evening and your time in Canada and everywhere in the world. So I will talk about life from the ice, cell viability after cryopreservation. You heard already a lot about cryo today. I just give you some information of data we need for my lecture, and I will skip those you have already heard. But it's very important to have this temperature scale all the time aware. Of course, the physiological temperature, melting point of water, eutectic temperature with water with saline solution, and then the glass transition temperature of water, which already has been mentioned several times. And then we have liquid nitrogen temperatures. The lowest natural temperature measured on Earth was -89 degrees centigrade, but there was in 2010 in the Antarctic a new record of -93.2 degrees centigrade. Why are we doing and why are we interested in these temperatures? You all know that you can store at low temperatures. You know that you have -20 degrees in your foot freezer, 4 degrees in normal refrigerator, or -5 degrees in the little freezer. Mean temperature on Earth is 0 degrees centigrade, the range we already mentioned. And now we have cryopreservation, and cryopreservation is below -80. But if you remember the last slide, we should be below -135 degrees centigrade, which is the glass transition temperature of water. And if we do the storage in liquid nitrogen, we are at -196 degrees centigrade. And we should take care of taking the temperatures in Kelvin as soon as we have differences in temperature. You already saw some of these pictures. It's always very good to learn from nature. We saw that the temperature in the world middle is 0 degrees. So we have a lot of plants and animals that survive deep temperatures, like the Canadian frog. You already saw this animal, but also the water bear is here, and other animals are also living there. This is what we can see in nature. Artificially, we have what we talk about today, cryopreservation, but always think of, is it maybe good to destroy cells and tissues with cold? Then you can use it with cryotherapy. We also have freeze drying, and we have hypothermia. And you probably all know, who are working with cells, you have cryofixation in the SEM and the microtome. So let's learn from these freeze-tolerant animals, for example, that we already saw in the first talk today. What can we learn from nature? By coldness, we inactivate genes, we reduce protein biosynthesis, pumps and ion channels will be closed, production of energy is reduced. In total, the usage of energy is reduced, some enzymes and genes are activated. And this is done in nature by using antifreeze proteins, for example, release ice nucleating proteins, changing the metabolism. We already heard in the talk from Gloria about the cryoprotective agents like sugars, polyol, and the DMSO that has been discussed in the last talk to a great amount. And the prevention of the cellular dehydration by the production and releasing of cryoprotectiva. So this is done in nature. Unfortunately, our human cells do not have this program, so we need to do something. I think I can now skip this slide, but you already learned that our cells upon freezing will shrink because water goes out. For the engineering students, I always have to mention that the cell membrane is only permeable for water and not the salt ions, which is very important. And yeah, you just have to learn it. And you have to learn that with protection, we can freeze our cells so that they are viable upon thawing. So this is what we want to do. To the engineering students, I also always say, excuse me, cells no longer age. And this is very important. And water is very important. We already heard a lot of water, glass transition of water, the change in density of water. Water is such a small molecule, but it has so curious properties that we have to take into account. And you all probably know that our biological tissues contain a lot of water. For example, 80% in the kidneys. But there was a question in the chat already about adipose tissue that I also wanted to freeze some years ago. And this has a lot of fat, but not so much water. So it's very important to look at the composition of the cells and tissues you want to freeze, because there are different reactions on it. And what we not already mentioned today is Arrhenius equation, because all our properties of our materials is dependent on temperature. And this is the rate of the metabolic reaction, which depends here on e to the power of -1 the temperature in Kelvin. So this is very important to know, but it's also very important to know that it is not only the metabolic rate, but you have to think of all the properties of materials that depend more or less on temperature. And if we go deeper and deeper with temperature, a lot of other properties may also change. And another, I think, important equation is Fantov's law. Fantov's law, there is a rule that he found out that when the temperature decreases in biological reactions by 10 Kelvin, the oxygen consumption decreases by half. And this is, I think, quite important. You see here, if we start at 0 degrees centigrade and say, let's say we have a lifetime of 1 hour, then at -20 degrees centigrade, we are at 4 hours. And at -100 degrees centigrade, we are at 1000 hours. And you see there are a lot of freezers at -80. And here you see 1000 hours of storage lifetime is not very much. But as soon as you go down to liquid nitrogen, then you have more than 100 years, which we do not need normally. in the field of regenerative medicine, but you see that storage at deeper temperatures gives much longer shelf life for your product. Yeah, now I'm an engineer, so the clinical requirements are different. For example, we want just to freeze human erythrocytes. Maybe you remember that you already saw the results of freezing red blood cells in the previous talks. Red blood cells need a very high freezing rate. Then we can think of stem cells, which is important for regenerative medicine. They do not need a high freezing rate. And then we have donor organs or tissue engineered organs we would like to store. Where is another problem? One is the freezing rate, and the other is geometry. It's very important to keep in mind that we have mass and heat transport. Organs have a large volume, could also be for blood donations. Blood donations have 500 milliliters, for example. Our chair, Jason Acker, will know very well. But our organs have a small surface. And this leads to, say, optimal freezing at the surface, but unfavorable freezing inside the organ. So in the optimal geometry is flat plates, or in case of blood, flat bags, but not what we normally have, cylindrical shapes of all the tubes that we use in cell culture, or balls, which is not very good for freezing. And so this is something we have to keep in mind, that geometry has a big impact on the outcome. In addition, as I already mentioned, the cells also have different geometries. I always like to show the sperm cells, which are really a different shape in comparison to other cells. And if we then go to tissue and organ, of course, we have this volume to surface ratio that is not very good for homogeneous freezing, which means the temperature should change homogeneously for every cell in the volume, and the freezing rate then also should be the same at every point. Now, if we do this freezing, you already saw this, we have one effect. This was this osmotic shrinking. As I always say to my students, we would like to have the healthy shrinking. If we freeze too fast, we have what we heard in the first lecture this day, the intracellular ice formation. And how can we see this, or can we observe this? Yes, we can. Here we see the intracellular ice formation in the cryo-SEM. These are red blood cells, and the white stuff is the ice. And here we are in the cryo microscopy, the cryo microscope, and we see the darkening of a heart muscle cell. As long as we know that intercellular ice formation is lethal to a cell, we can say, oh, probably these red cells did not survive. Actually, I did this freezing and I wanted to look it like this, to have a nice picture to show this. And here this darkening is also something telling us that this cell will not, to let's say 80%, will not survive upon thawing. So we can visualize this. And you saw also some other pictures so far today. Excuse me, here we have yeast cells, and here we really see in the light microscope the change in volume, in shape. So this was freezing, the shrinkage upon freezing. And here we see renal cells, human renal cells in cell suspension. And please have a look here. This is minus 22.6 degrees Celsius. Then we go down with temperature. You see extracellular ice formation. You see the change in color, which gives another optical signal. And here, just 1% more, also deeper temperatures. Here you see one of the cells has intracellular ice formation. And this can be seen in the Cryo Microscope. And yeah, all these findings have been put together by the leader in our cryobiology field, Peter Mazur, and he put this together, and you already saw this, this shape, this U-shape on top, saying that we have much cell damage if we freeze too slow, and that we have a lot of cell damage, intracellular ice formation, if we freeze too fast. And there is a cooling rate that is optimal. Please do not find this one optimal cooling rate. Please find the range of optimal cooling rates, because if you remember, I told you that in organs and bigger volumes, it's not possible to have homogeneous one cooling rate. It's good to know the range that leads to good survival. And so it's nice to know this range of cooling rates that is favorable for my cell type, for my tissue. Now, we did a study in this area. We did a study with I would say, probably simple skin cells, keratinocytes. And we measured membrane integrity after thawing. We used different DMSO concentrations. I only show you 2.5% DMSO results in this picture. We did up to 2-stage cooling rate protocols. So here from 4 degrees centigrade to -30, we had cooling rate number 1. And from -30 to -80, we had another cooling rate. In some cases, both were the same. Then it was a 1-stage cooling. And the colors now show you cells with intact membranes. Here, in this little area here, we have intact cell membranes for more than 90% of the cells. We also changed the DMSO concentration. So by increasing the DMSO concentration, if you remember the last talk, unfortunately, membrane integrity goes up. So DMSO helps for membrane integrity. And it also lowers down the cooling rate. Excuse me? The cooling rates. Unfortunately, this diagram with 5 or 4 different DMSO concentrations took us 1 year, because we only had one freezing device, controlled freezing device, to do this. So this is a very expensive result. But what I want to show you is that a little deviation of the cooling rates drastically change the cells with intact membranes to near 30%. So it's very important to keep your cooling rates in your cell volume, in your tissue engineered construct as homogeneous as possible to the optimal rate. And then there is another important feature we have to take into account. If we cool down, here we have temperature over time. If we cool down our solution, this is the cooling rate. We do supercooling, which means we are below melting temperature. And then statistically with our water molecules, the first ice crystals build or are built, and then the latent heat is produced. This means that all our cells who have already been at this low temperature go up again to melting temperature until all the cells have the same melt temperature again, and then we go down with the cooling rate again. So we have to keep this as small as possible because our cells don't like this going up and down with temperature during this process. Okay, I forgot the animation. Yeah, and the result that we already know is that survival rate versus cooling rate depends on the cell type. And as I mentioned in the beginning, erythrocytes like to have high cooling rates of more than 3000 kelvins per minute, which we cannot realize in a 500 ml blood bank. Technically, it's not possible to get rid of all this heat in this short time. Good. Now, how do we know whether our cells survive or not survive our freezing and, of course, thawing procedures? For this, I would like to show you some of these methods, and a lot of them you have already heard about in the talks before. So let's start with the Cryo Microderm. What is this? The Cryo Microscopy already mentioned, and then we have cell counting. I mentioned the cell membrane integrity that Gloria already discussed in her talk, and go up to the epigenetic effects that we already mentioned in the talk before. So Cryo Microderm is quite nice that we are able to freeze sections in very thin. Here you see in the thickness of 0.5 to 100 microns, very thin slides of the frozen tissues, and then we can do a lot of investigations, and we do not have to do the paraffin section. Cryo microscopy has already been mentioned, that we can visualize the freezing process in the microscope. Here you see some cells, and here the growing ice to the cells. And here again, you remember this is the hard muscle cells in the ice. And these were mesenchymal stem cells that we froze and visualized. It is interesting that in the cryomicroscopy, you could also do single cell tracking. So this is something that we did in one of our studies, that you can really track the cells. And you remember the intracellular ice formation can be seen by darkening. And so we were able to track the darkened cells. And then we did the thawing, of course, and the viability testing. And what we found out is that not all the dark cells were dead. It's a very nice method to really see what happens during freezing, although of course this is in a thin layer of the cells and the cryo microscope and not a real, of course, 3D volume that we need to freeze in daily life. But you get a lot of information and you can really see what you do with the cells. We mentioned before the DMSO. The DMSO, that is the CPA that goes into the cell. And there are also cryoprotected agents like sugars with higher molecular weights that do not enter the cell, but you can try to get it into the cell by electroporation. And all these things you can really visualize. Yeah, and then we have the cell counting, or you already heard about the dyes, the trypan blue staining, the fluorescence staining, the automatic cell counting. Here we have image analysis picture from the Y cell, where we get information. The number of the cells, the number of living cells can also be detected. Cell size, of course, so we can visualize the shrinkage. We can see this. I'm always a little bit concerned about the dyes, whether they really do not change any properties of the cells. So I really like to find other methods, not to have to dye the cells. So try to do the reproduction cycle after thawing. Yeah, with the Coulter principle, we can again measure the number of cells, the cell cycle size distribution, and viability is also possible to be measured. The nice thing with the Coulter counter is that the principle is the change in electrical resistance here, and this change in electrical resistance is proportional to the cell volume. And this is very easy to measure. You just have to know the geometry of your cell to get from the volume the cell size, really. And not all cells are balls. Suspended cells often have this shape thus. The other assays, cell assays, were the ones that we already heard about, the MTT test to see viability, Alama Blue, the metabolic testing. As a simple engineer, I know the green cells are the live cells and the red cells are the dead cells. Here we did an apoptotic cell assay. So the orange parts are apoptotic cells, and this is a frozen kidney, and we sliced it with the microtome, and we looked at the slices, and we looked how the cell survival is, and we are happy that we have green areas here, unfortunately not in all depths, and this is due to heat and mass transfer, which was not 100% optimal, because in the optimal case, enough cryoprotectant would have been diffused into the middle of the tissue, and also the release of energy would have been more homogeneously. So this is in direction of tissue engineering and organ preservation to get this information. And we also see these results here. Actually, I only show these results in teaching field because this is a cross-sectional area through a frozen tissue. And what you can see here, the outside is green, so everything is fine. Heat and mass transfer was fine, enough CPA, temperature was fine. Here we have red cells, and probably because of impaired mass transfer, so the CPA was not here. And it's very important to know the coefficient of diffusion of our CPAs, because it takes quite a time for the CPA molecules to diffuse into the tissue. You know, yeah. Then we started a little bit with epigenetic tests, and from the epigenetic tests, we learned that now, when we do this freezing and thawing of our tissues, that we do a 24 hours recultivation, and we do measurement of metabolic and reproductive assays after this 24 hours. Because we realized that directly after thawing, there are some biological processes, negative processes, that are reversed, unfortunately, after 24 hours. Yeah, so we started to do this epigenetic testing, and we saw Anamika Chatterjee's publication in this talk before. We took the cord blood cells from the stem cells from the cord blood from this monkey. We froze it, and then we did the measurement of the histone modifications and the DNA methylation, and this with DMSO, with glycerol, and we have the controls. And unfortunately, as DMSO is known to have an effect on the epigenetic, which then has an effect on the differentiation of the stem cells, could be shown. It was not in all the cases, so it's very important to our knowledge so far that upon thawing, after 24 hours of recultivation, you should do these, the measurement of changes in epigenetic to be sure that your protocol for your specific tissue and cells really does not do any harm to the cells. So this is what I wanted to discuss this evening with you, more from the view of a process engineer. A process engineer that looks for viability of cells in the healthcare by cryostorage. But we know that cryostorage is also important in the environment and the economy. We looked at these influencing parameters, water content, cooling rate, thawing rate, which should be homogeneous in the volume. Geometry is very important. Of course, cell density, cell numbers in the volume is important. Cell cycle, I did not mention this, but this is of course also important that it is not negatively impaired. Viability after 24 hours after recultivation. Look for the epigenetic changes to really be sure about your protocol. Try to reduce DMSO. So we are now at the stage that we only use 2.5% DMSO. Of course, we try to develop with electroporation methods so that we do not anymore have to use DMSO. And please be aware of heat and mass transfer and about the impact of deep temperatures on these properties, like the coefficient of diffusion. And with this, I would like to thank you for your very kind attention, and my PhD students who did all the work with the freezing of the cells. Thank you so much.
Great. Thank you, Professor Glasmacher. In the interest of time, we're going to move right into the next speaker. If you do have questions for Professor Glasmacher, please include them in the Q&A, and Professor Glasmacher can review those and respond as we're going into the next session. So it's my privilege to introduce our next speaker, and I do apologize that I'm probably going to mispronounce his name, and I've already pre-warned him on that. But Dr. Dennis Pogozia from the Hannover Medical School in Germany will be speaking on the long-term biobanking of multipotent stromal cells, and specifically introducing us to some very exciting work that they've published, I suspect published a few years ago, looking at the impact of temperature fluctuations on this specific cell. So please go ahead.
Okay, thank you very much. Yeah, so it was really great and very important talks today about a lot of very important practical aspects on cryobiology and cryopreservation of basic principles, and also on particular on preservation on different cells. I will go more specific on what we have in particular experience with, and we will talk also on challenges of practical biobanking of multipotent stromal cells, which is also related to future perspectives or current perspectives also in clinical application of multipotent stromal cells and stem cells in general. Yeah, and in particular with our research, we are very interested in placental material. So here I will show you why is it important, or why placental material in general. There are a lot of clinically active derivatives which can be derived from placenta, which include, for example, starting from placental extracts and tissue lysates, also cord blood serum, and we are interested highly in placental MSCs, and there are also placental tissues and amniotic membrane and chorionic membrane, which is already used in clinics, in particular in the field of ophthalmology. Very important and very unique property of this temporary organ is that it contains fetal and maternal surfaces, which are patient-specific, provide patient-specific material for two individuals. Yeah, fetal surface has cells which belong to the fetus, and maternal surface consequently to the mother. So we have from this source two patient-specific material. There are a lot of advantages of placental-derived material, like there is very low to no ethical concerns with it, really highly available source. Yeah, everyone who is born, he has placenta. Also, what I just mentioned, possibility to get autologous material, and moreover for two individuals. It has immune evasive properties. I will talk about this a little bit later. Yeah, it has low expression of major histocompatibility complex, and also there were a lot of reports that on cellular therapy that there is no tumor formation, which we usually see in some other preclinical studies, like with iPS cells, for example. And placental MSCs are also highly plastic in terms of differentiation. There are some works where it is shown that they not only differentiate to classical adipose, osteo, and chondro lineages, but also to the other lineages, and also others. And we showed really high proliferative capacity of placental MSCs in comparison to the other sources. Yeah, about the antigens on the cell surfaces I already mentioned. Yeah, so there are a lot of natural structures which can be derivatives for relevant placental derived materials. For example, cord blood, umbilical cord itself, then amniotic and chorionic membrane, and placental tissue. And it's really a lot of material, which gives us a lot of things to work with. And also placental MSCs can be used in tissue engineering to generate bioengineered structures. Yeah, I can talk about placenta and how great it is, like, for hours, but we are limited today in time. And a couple of years ago, we were invited to write a review about it, and really like history of application, of clinical application of placental material, and a lot of studies related to it. It's over 100 years. So I put here on the bottom PubMed ID. If you're interested here, it's a lot of information which we included, and it's an open access paper, so you can have a look if you need some more information about it. Yeah, so in particular to this talk, we're interested in MSCs of placental origin, and also we did characterization of placental MSCs from the amniotic membrane and from placental tissues, and compared with MSCs from the bone marrow. And we did it also in comparison with non-human primate model, common marmoset. It's like a really small New World monkey. A lot of clinical trials which are advanced, so to say, have to move from a traditional rodent model, yeah, because it's really far away from the human, and application of primate model is much more relevant at the late clinical trials. So we showed also placental stem cells in comparison monkey with the human, and in particular what is interesting from this paper is comparison with the bone marrow, and we showed that placental MSCs in comparison to bone marrow, they possess much lower MHC class 1 expression in the primary culture, which increases during passages, but also at the beginning for up to 10 passages, it's really low. Yeah, and they are capable to differentiate to classical lineages, and we can culture them much longer in comparison to the bone marrow MSCs. Yeah, anyway, if we want to go to clinics with our stem cell products, biobanking is inevitable. It's really like an inevitable step for the clinical application of stem cells in regenerative medicine. It permits us, it gives us a lot of benefits. Yeah, it permits accumulation of sufficient amounts of material that are necessary for transplantation or for other therapeutic approaches. It also permits us interrupt continuous cell culture maintenance, yeah, because cells, especially primary lines, they get exhausted with culturing and with passaging, and also it's a lot of additional costs and materials and so on. And cryopreservation permits us to stop it and to reactivate it when we need it. Also, it permits us targeted cell banking. Yeah, we can, for example, store targeted patient-specific material, which can be used in the patient in many years or even in decades. And also, it gives us time for necessary tests, yeah, to perform different pathological tests and contamination and so on. Stock management and logistics, of course, yeah, when we need to transport somewhere, it's better to transport them in the frozen state. And also it gives us what is the most important, availability in a ready-to-use manner. We can take them when we need and apply them when we want and where we want. And some other advantages which we all know about it or can think about it. But clinical application, clinically relevant stem cell biobanking has a lot of requirements, which also have been partially covered today. I will just briefly go through them. So, of course, we need to have high levels of survival after cryopreservation. Yeah, that most of the cells which we preserve, they should survive. And the more the better. Yeah, and they of course have to be preserve the phenotype and the functional properties. What is very important for stem cell biobanking, they should be compatible with GMP and GLP standards. Yeah, because a lot of protocols, they contain, for example, animal serum or some other xenogenic products, and this is it cannot be applied. It's not allowed to apply in the patient. Also, it should be a possibility for long-term storage. Yeah, like for example, Birgit mentioned, in the -80 fridges we cannot store them for so long. Yeah, but if we need them in years or decades, this is very important issue, and this is what I will address in the further talk. And there are, of course, debates about the reduction or substitution of DMSO as cryoprotectant. There have been a lot of nice information covered about it. Yeah, and Dr. Elliott also mentioned about the balance between toxicity and effectivity of cryoprotectant, which has to be taken in account. Yeah, so this is all very important aspects. In particular, with biobanking of placental MSCs, I would say they are quite easy and robust cells to cryopreserve. There have been a lot of works already for many years, and you can preserve them very nice with viability rates over 90%. We also worked a little bit in this field. We also investigated possibility of hypothermic storage and with alternative cryoprotectants and so on. This is also open source paper. You can have a look if you need more details, or you can ask me later if you need more details of it. Yeah, but very important also, besides just suspensions, bioengineered constructs on MSC-based products. This previous speaker, Professor Birgit Glasmacher, her group has excellent experience about this and great expertise. We worked also together on a lot of these projects, and in particular we adopted this technology with placental MSCs with some really nice results, and also we looked at possibility to cryopreserve these constructs. Together with our German group, together with our collaboration partners in Ukraine, went further also to cryopreserve tissue engineered construct and also placental tissues, yeah, and natural structures. And we had a lot of objects where we studied this, like placental explants, umbilical cord explants, and fetal membranes, but also from the tissue engineered, it was spheroids and alginate microspheres, similar like we did with Birgit. Yeah? Also, this was really large work for several years. We did a lot of Cryo Microscopy and other studies and characterization. It's also a topic for another talk, so I will not stop about it here very much. But what is very important here is that from cryopreserved placental tissues, amniotic membrane, different parts of placenta, like placental cotyledon tissues, amniotic membrane, and umbilical cord, we were able to isolate after cryopreservation viable cells, which completely retain their phenotype. This is it's not cryopreserved cell suspension, but it's really cryopreserved tissues, and after freezing, we were able to isolate the cells. So all this work has been performed for several years, and during these years, we and our partners who worked on it, we noticed that while we have the same primary culture, at different points of the research, we have different viability rates, and sometimes cells look better objectively, sometimes worse, and behave a little bit different. And then we had an idea that what happens to it. Yeah, there is a lot of things going on in the lab, and you open the fridge, close it. Yeah, a lot of things can go on also during practical biobanking in the long term. Yeah, sometimes it can be just stock management. You take out sample, put it back. Sometimes it can be some accident or some transportation where you can temporarily, or maintenance also, where you can temporarily store samples at -80 instead of liquid nitrogen or -150. And during long-term biobanking, for example, with placental cells, when they are derived from placenta at the birth of a person, and they can be used later in decades, if in many years, if not decades. Yeah, then such temperature fluctuations in sub-zero range, I'm not, of course, talking about interruption of the cold chain, but in sub-zero range, such small temperature fluctuations, they can really accumulate with time. Yeah, in many years, somebody opened the fridge, took the sample, moved the sample, just stock management and stuff. This can happen. So we decided to study what happens to the cells, and we tried to mimic these temperature fluctuations, mimic long-term biobanking by temperature fluctuations during cryopreservation. We took for MSC's really standard protocol, first 1 K per minute to -80. Many of you know Mr. Frost is into liquid nitrogen. This is widely used everywhere for stem cell with normal cryopreservation medium. And we made repeated temperature fluctuations from the liquid nitrogen to -80, from liquid nitrogen to -100 and to -150. And we did a number of cycles, 5, 10, 20, 30, up to 50 cycles of this temperature fluctuations, and try to see what happened with the cell. We measured directly in the sample with thermocouple. We measured directly the dynamics of temperature changes. So here you can see some differences with it. And also we performed Cryo Microscopic analysis. So we froze, you can see in both like this. Yeah, we froze down to -100. It's here on Cryo Microscopic studies, it's only below, only until -100 degrees. We just wanted to see structural changes in the ice, but our device did not allow to go below -100. And then we warmed up from -100. We warmed up to -7 degrees, and we saw how the ice crystals change. And then here we also did the temperature fluctuation. So at the first image you see samples at -80 degrees were at the first cycle of temperature fluctuation. So from here we start, yeah. Then samples at -100 degrees at the last cycle of temperature fluctuations from these 50 cycles, and -80 at the last cycle. You see with arrows, I don't know if you can see it very good, slide. So generally it looks everything okay, but we were able to see some really small modifications, which I will talk about it a little bit later. So then we thought the cells, and after each cycle also we thought part of the cells, and we did a different evaluation of their viability and other parameters. The simple cell count showed that temperature fluctuations itself from liquid nitrogen temperatures to 80, 100, and 150 different ranges. They do not influence so much. This range does not influence so much as the number of cycles, but after 30 cycles already we can see significant changes. And then we did classical cell survival test with 3-band blue staining, and here we already see that the range also of temperature fluctuations, not only the number of cycles, but also the range between the fluctuations, already from the 10 cycles, it has significant impact. So if we those cells to -80 and freeze them back to liquid nitrogen, already you can see significant impact on the cell survival. Yeah, and of course, very important functional test for mesenchymal stem cells. It's their capability to adhere to plastic, and we did adhesive properties. This is what Glyn have mentioned, that it's important to test them after 24 hours. Yeah, for example, then you already have a real idea what's going on. Yeah, and so we saw the compromise of adherence depends also both on the range of cycles as well as on the number of cycles itself. With MTT test, it's a very informative test which shows metabolic and proliferation activity after these procedures. And here we see great impact on the range right away. Right away, you can see it, yeah. And then also accompanies the number of the cycles, yeah. And also we did a functional test for MSCs, the differentiation in adipogenic, chondrogenic, and osteogenic lineages, and visually everything looked fine and everything looked the same. But we did really big microarray assay of more than 40,000 genes, and we compared already after differentiation, and we checked genes associated with adipogenic, chondrogenic, and osteogenic lineages, and here you can see tenfold up and down regulations we observed, and tenfold and more they are marked in red and green down regulation and up regulation of more than 40 genes we were able to observe it. So this is also very important to consider, and this is also maybe has to do what Birgit was talking about epigenetic changes and some modifications. Even though they differentiated fine, but you can see it's like really significant changes. Yeah. So also on this, as Jason already mentioned, we published a paper some years ago, and a lot of details can be found. Further details and also methodological approaches can be found in this paper. A very important summary from this. It's a lot of text, but it's a lot of important information that we have to consider. So we see significant changes in the ice crystal and channels during cryo microscopic studies in the range above -40, and temperature fluctuations below -80, there are very slight changes in inclusions and in transparency of the ice crystals, but not so obvious as at the high temperatures. But the total number of cells, viability, and metabolic and apoptotic parameters, they did not significantly differ from controls with temperature fluctuations in the range in -906 to 100 in less than 20 cycles. But when we increase, as I already talked, the number of cycles and as well as the range, it leads to significant lowering of the number. Yeah, and also adhesive properties are compromised, and this has to be considered also during practical biobanking. So the main take-home message here is that temperature fluctuations in the samples typically inherent to practical biobanking in these scenarios which we described. It should be considered during long-term storage of placental MSCs and generally of stem cells. And it should be especially deliberated for the prospective clinical application in the field of regenerative medicine. But development of strategies to avoid such temperature fluctuations is quite feasible and should not be overlooked. Yeah, there are a lot of new robots developed, and a lot of different biobanking strategies from the point of view of how to handle and to how to maintain that such temperature fluctuations are avoided completely, or at least as much as possible. And this is really important. Maybe not so much to the lab, but we see also a lot of difference in the lab. But if we go to clinics, this is really important to consider. And I just, as additional information, this is not related to the cells, but also to placental material in particular in ophthalmology. This is just not some theoretical deliberations and speculations. This is really important already in the clinical applications. We also collaborated with German Society for Tissue Transplantation, and last year we studied influence of repeated freezing procedures on amniotic membrane, on placental amniotic membrane for application in ophthalmology. Yeah, in this case also we saw some changes and also gave some recommendations. So thank you very much for your attention. Here's also a brief overview of the topic which we discussed, and some links if you're interested in more detail in this topic. You can have a look, and also you can contact me and ask me. Thank you very much.
Excellent. Thank you for that very insightful talk. In the interest of time, I'm going to defer the presentation that I was going to give, which was really going to provide some additional evidence around the impact that transient warming and temperature excursions had on hematopoietic stem cells. I will include a couple of those slides in the talk that I will be giving on June 2nd as part 2 of this workshop. So if you are interested in hearing that, please join us on June 2nd. So with that, with the last few minutes that we have available for today, I would like to open it up to any questions from the panel or from the audience and the attendees. You can use the raise hand on the bottom portion of your Zoom window, or if you're uncomfortable, please put your questions in the Q&A box, and the panelists will have a chance to look at those and either respond in real time or after this call. So if you do have any questions, please just raise your hand. And I'll just wait to see if there are any questions from the attendees. For those that haven't been following the Q&A section, there have been some great questions that have come up, and many of the panelists have answered them. So don't just look at the open questions. Please go in and dive into the answered questions, because there is a lot of insight that has been provided by our panelists. All right, so Dr. Pogazia, there's couple questions for you, and maybe I'll just, if we can answer the question from Dr. Leah Marquez-Curtis. Her question is, MSCs from various sources are currently explored in clinical trials as potential therapies for COVID. Have you considered your placental-derived MSCs for this application, and what do you see as advantages over MSCs from other sources?
Yeah, thank you very much. We particularly don't apply ourselves. It's in clinic. We do more the research, but I see the great advantage of this material, of course, first of all, because of its high availability in comparison to the other sources. Yeah, it's to obtain this material, it's not a problem. Yeah, and also the opportunity to obtain sterile material in the case of C-section is possible, and a lot of great advantages, which include high proliferative potential of the cells. If you get MSCs from the other sources, sometimes they grow 2-3 passages, maximum 10, and then they start to degrade or stop growing. Placental MSCs, of course, it depends on the part of placenta, but we were able to culture for continuous culture for over 100 days and up to 20+ passages. Yeah, this is a great adventure. Of course, what we just talked about is this patient specific for 2 individuals, but also they can be applied for in autologous setting, because they have low expression of major histocompatibility complex, which is a great advantage for clinical application in this case.
And then maybe I'll just follow up with another question was asked to you. Are there any ethical regulatory difficulties to working with placental tissue in Europe?
As far as I know, there are, of course, regulations, but placental tissues already applied in clinics, very widely applied in the field of ophthalmology, and it's not only in Europe, but it's worldwide. And in the other countries, the range, of course, in Europe, it's very strict regulations, and they're not only general European, but also specific for the other countries. So it's very important to deal with them. In the other countries, application of this material is also very broad, and it's readily applied non-cellular material in the form of extracts and lysates, but also tissues and cells are successfully applied in clinics for decades.
Excellent, thank you. So if there are any other questions, feel free to reach out to the panelists. I think many of us have provided our email, or through the Stem Cell Network, we'll be able to get back to you. And with that, I'll turn it over to John to just give us a wrap up and a lead into the next part 2.
Yeah, thank you very much, Jason. And thank you to all of our speakers and all of our chairs as well for the fantastic information they provided through the talks and contributing and volunteering their time for this workshop. So just a reminder that our next scheduled part of this workshop, it's a two-part workshop, will be on June the 2nd, Wednesday, June 2nd, and it will start at 12 noon as well. If you have any issues with your links, your Zoom links to access this webinar, please do contact me. You'll have my email address through the registration process. So with that, thank you to everybody and see you next time.